You are now live.
Thank you. I don't think Ian Donaldson's here though.
Oh, sorry. Yeah,
oh, no. There he is. Sorry. Yeah, there you are. Okay. Thank you.
Brief interruption. So
that's okay. No worries.
Okay. So good afternoon, everyone, and welcome to the scoping call on algae and marine feedstocks. Supergen Bioenergy Hub are holding this afternoon. So can you move to the next slide, please, Dan?
Yeah, it's Emma, actually. I'm moving the slides this time, so there we go.
So the agenda this afternoon is going to include an introduction to the hub and the themes within that Patricia Thornley, Director of Supergen Hub, is going to give. I was going to give an update in terms of what the activities we're doing on feedstocks within the hub, and then we're going to hear about the particular flexible funding challenge call that's coming up from Emma, and then we have a series of presentations from researchers within the community on algae and various activities that are happening now, and also could help inform what could be within scope and the challenges that are there at the moment. We'll then have an opportunity for a roundtable of discussions, including ideas for helping to scope out this call, and then we should be finished by 3:30. So first of all, if I could hand over to Patricia.
Okay, good afternoon, everyone. Can we have the next slide, Emma? Okay, so what I want to do this afternoon is to give you a very brief introduction to the Supergen Bioenergy Hub. I'm sure that many of you are familiar with this already, so I will try to keep it brief and to highlight really the bits that are relevant to the call that we are scoping this afternoon. So the hub exists as an EPSRC, BBSRC jointly funded mechanism to bring together academia, industry, and wider societal stakeholders in the UK to focus on sustainable bioenergy deployment. In order to do that, we divide our work into 4 key areas. The first of those is resources, and in some senses, our topic group on resources, led by Ian Donaldson, whom you've just heard from, is the most relevant to the call this afternoon. You would think that algae, being a biomass feedstock, would therefore sit wholly within resources. And that's why we've asked Ian to chair this afternoon's session. So from a Supergen perspective, what we're interested in when we look at resources is looking at options for feedstock production in the UK to support UK bioenergy systems without negatively impacting on food production and maximising environmental and climate benefits. So that all seems fairly sensible, but let's look at the next topic group 2, which has 3 leaders, Chris Hardacre in Manchester, Tony Bridgewater at Aston, and Jason Hallett at Imperial. And together they work to develop new pre-treatment and conversion technologies that support the hub's vision for UK bioenergy. Now, I'm going to suggest that while algae is a feedstock and is important to us for topic group 1, actually some of what comes within scope of this call might move through into conversion of that feedstock, and particularly perhaps the pre-treatment elements of that, the first stage of conversion techniques. The third topic group in Supergen is vectors. That's led by Marcel McManus at Bath. And within that, we're looking at what do we convert biomass into, which is useful to us. Now, the obvious answer is we're converting it into energy. Well, what form of energy? What demand is it fulfilling? What is it displacing? Because all of those are key around the reduction and environmental credentials. But there's something a bit more to it here when we think of algae, because one of the main challenges with algae systems is making them financially viable and extracting the best value from the raw material. And so when we look at vectors, we don't just look at energy vectors, we look at bioproducts as well. And so it may be that some of what is funded within this call might look at particular algae feedstocks in order to produce particular bioproducts, which are perhaps unique to the algae domain and inhabit the biotechnology landscape in that sense. So the vectors theme could be very important here. And then when we do all of that, one of the things that we love doing in Supergen is systems analysis. We bring all of this stuff together, the feedstock, the resource, what we're turning it into, and we look at what the net impact is. Because if it isn't maximising the affordability, resilience and carbon performance of our energy system, then it isn't worth doing. That's what we were set up to do, and that's what we have to deliver on. So no matter what you're writing as your proposal for this call, reflecting on how you're delivering against that trilemma and delivering wider ecosystem benefits is a really, really important thing to do. Within Supergen, we deal with the systems work via our case studies. And if you look at our website, you will find a report there which details which case studies we've chosen to focus on. And my recollection is that there is at least one in there that specifically focuses on algae. So next slide, Emma. So that's how the overall work sits within Supergen. We've talked about resources, pretreatment and conversion, vectors and case studies. But let's not forget that there's a point to doing all of this. And what we're trying to achieve, and one of the key things about this call, is to try and improve cooperation. You'll see that down in the bottom left. We're trying to bring in new people, new partners to get new insights and to get new people involved with this. And we're trying to improve the cooperation and coordination between partners. We need to, of course, integrate the science and engineering so that it all makes sense. But also we want to improve our industrial understanding of what might make sense. And I think this is particularly important for the algae feedstocks because we need to understand what the potential market is and who's going to deliver on it. Our early career researchers are really important to us in Supergen, and we provide training for them and a national network, and it would be lovely to see some of those early career researchers actively involved in proposals against this call. And finally, I mentioned the impact word down on the bottom right. We love having engagement with industry. There are many of them you will see in the next slide who have a formal relationship with us and many others who have informal involvement in a whole host of our projects. We are extremely involved with the policy regime on this. And for the algae work, I think that BEIS and DEFRA are probably the main two in this context. And I think that we mustn't forget the importance of societal engagement around this. And this is particularly sensitive when it comes to algae, because I'm sure that many of you know that the wider public have a huge attachment to our coastal regions, to our shores and our oceans. And so they want to be very confident about what's going on. For those who haven't seen it, the UK Climate Assembly published its report on what a cross-section of the demographic of the UK population thinks about our different options for hitting net zero by 2050. And one of the interesting things about bioenergy in that was it was the technology that recorded the highest level of uncertainty among the public, the one that people didn't understand, didn't know about, felt it all depended and there were too many options. So I think it would be great if some of what was done in this attached on to our wider efforts within Supergen to engage with the public and to connect with the wider societal aims around this, as well as just the research element. I think that's it, Emma, just the next one. Next slide. Yeah, so a reminder of some of the people who are already involved, but of course the point of this call is to get some new people involved. So don't focus too much on this, and let's move forward with trying to find some new ideas, new projects, new people, new companies. Back to you, Ian.
Right. Could you move on to the next slide, please, Emma? Or are you?
I've got my
next,
sorry. But I don't. Hang on. Oh, did you send any slides, Ian?
No, there's just one slide. That's fine. So it was really just okay. So I can talk about. No, that's fine. So I'll just give you. I guess the important message here really is just the fact that we don't have any activity really in the main part of the hub on algae. And I was just going to give a brief description of what we are doing, which is really around mapping land marginality and the productivity, in effect, of land-based bioenergy cropping systems. And so that I guess within this call, it can people can think about how that interacts with what we're doing. But really, it is about thinking about the marginality of land and how that we can obviously target the cultivation and the accumulation of biomass from those areas which are least likely to interact with food production. So obviously, the case of algae is an alternative way of looking at that. I guess we're looking at biomass productivity and how can we optimize that, and particularly thinking about the establishment phases, which tend to be the periods of higher risk, and also thinking about future climate scenarios, so impacts including extreme weather events. And then there's a range of cross-cutting themes in what we're doing, so around land use change, particularly thinking about the sort of soil carbon, greenhouse gas emissions and biodiversity. And that interacts with both previous and other previous Supergen activities and also other activities that are taking place. Also very much as Patricia was talking about, thinking about matching biomass quality to end uses, and also within the context of the sort of systems context around the sort of negative emissions and net zero. So that's how we're looking at the moment in relation to feedstocks within Supergen. So there's sort of modelling around that, in particular around around marginality and productivity of biomass, and what that might look like in the future. But the important bit within this call is trying to think about, or as an area, as a potential challenge area, is around the opportunity to look at the algal, and either from a macro or a micro perspective. So Emma, I'll hand back over to you then in terms of describing the flexible funding calls themselves.
Okay, thank you, Ian. So my name's Emma Wild. I'm the Supergen Bioenergy Hub project manager, and I'd like to talk to you today about the opportunities that we have for flexible funding within Supergen Bioenergy Hub coming up in the next couple of months. So we do have various flexible funding opportunities. The first one, and the one that we're talking about today, is a flexible funding challenge call for which will be launched in October. And we are anticipating funding 2 projects of up to £100,000 each. The call is going to focus on 3 areas. So we've got anaerobic digestion and substitute gases, global land use change, and algae and marine feedstocks. And we've already held the 2 previous webinars on AD and global land use change in the past couple of weeks. And this is the last one. And then we'll be using the priorities that we've identified throughout these webinars to redefine the call and launch it next week on the 5th of October. In October 2020, we also plan to launch a call for 2 Supergen fellowships, and we will be having an introductory webinar for that on the 14th of October for anyone who's planning on joining, and that's aimed at early career researchers We did try to have a call for Supergen fellowships last year, which wasn't successful. So we are accompanying the webinar. We're having a webinar to try to help people prepare their applications and to try to direct them better so that we know that they'll be targeted in the right way and we'll get some better quality applications. Within Supergen, we also have rapid response funding, which is available all the way through the project, and generally for kind of small projects of up to £25,000 each with industry, and they're aimed at carrying out a high immediate impact research, which can be funded straight away. And in terms of eligibility, all Supergen Hub members are eligible to apply for any of this funding. And if you're not sure about whether your institute is a Hub member or not, please contact me and I'll let you know. Sorry. So the challenge call itself, as I've said, is going to be focusing on 3 topics and will fund 2 projects of £100,000 each and address the 3 areas. We're identifying the main priorities for each of those areas during these webinars. All proposals are expected to be collaborative with at least one other institution and to align to the objectives of the Supergen Bioenergy Hub. We expect there to be significant engagement with the Hub, and we expect also to have presentations from you on your progress, results and the impact of the project. All proposals should deliver impacts, and we need to report these to the research councils because this is research funding from them and we need to kind of quantify that. So, oh, I had one more slide, I think, but it's not here. So there's an outline timetable, which seems to have disappeared here. We're going to release the call on the 5th of October, and assessments will be made in, I think the closing date is going to be the 30th of November, if I remember rightly, and in January we'll be making the awards, but I will make sure that's updated in the slides that go around to everybody after the event. Thank you. I'll hand back to Ian.
So thank you, Emma. So we now have a set of presentations to talk about what's currently happening and in the UK on algae, and also to flag up to help us with the discussion at the end in terms of what might be some of the gaps and research targets and challenges that are there as well. So the first of the presenters this afternoon is Michelle Stanley, who's at the Scottish Marine Station. So I'll hand over to Michelle now.
Thank you, Ian. Could we go to the first slide, please? Thanks. So I think this slide was produced about 8 years ago, but is actually still very relevant, and it just sums up where we sit, where seaweed or macroalgae is concerned. Where macroalgae, when we look at it, we classify it due to its colour, so brown, green or red. I've worked on all 3 for my sins, and we look at it from anything from where we're putting inputs in. So say we're looking at on-land tank cultivation, so wastewater, artificial fertilizers, or if we're looking at sea cultivation. So a lot of the work we do up at SAMS, we have 2 seaweed farms. We look at sea cultivation. Where we are concerned, we don't actually use artificial fertilizers. However, in other countries such as China, where they have large scale production, they will use artificial fertilizers to help production. We have cultivation goes from everything from what we would describe as natural blooms. So if it's a slow news week, usually in the summer, you'll have news on about the Brittany coast in France, and the fact that every year they have about 60,000 tons of a green seaweed called Algae washed up on the beach. And this is because it blooms because of the nutrients that are being put in this case, usually from farming. We have what we would describe as standing stocks. So that's wild material. And for those who were looking at this last year, a company wanted to harvest in Scotland standing stocks for alginate production, and because of how things were handled, and this is where social science and social license comes into play as well, because the science wasn't really there, but because of public feeling, the Scottish government have now banned the wild harvesting at scale of 5 different types of kelp or brown seaweed species. On land tanks, some work we've done with a particular red species called Pepperdullis, which is used mostly for the food market, but actually contains also antivirals and anti-inflammatories. Patricia briefly touched on conversion technology pre-treatment. A lot of the pre-treatment work usually involves acids, as it would for other forms of biomass. There's also some work going on with enzyme pre-treatment, and actually there's a bit of flexibility here, or there's a need to actually expand what actually happens in this area. When we get to products, we're still into biomethane, bioethanol, butanol, platform chemicals as well, things like succinic acid, lactic acid, and we also have thermochemical products. And markets, again, everything from cosmetics, pharmaceuticals, specialties, biofuel, bioenergy, and as I said, bulk bio-based chemicals. So if we go to the next slide, please. This just gives you a bit of an idea of where we're going, where seaweed's concerned and seaweed farming. This is a diagram of our main farm. And I've got a link at the slide at the end for Sam's YouTube channel, and we just put up a new video on the work we do with seaweed. Where we're going, and this is in aquaculture in general. So seaweed sits in this odd place. So we sit sort of as a plant, but we're not a plant because browns aren't considered plants. And we're considered by the license, so we have a license for this farm. We're considered as aquaculture, but then the main aquaculture companies don't consider us as aquaculture because they're more interested in producing things like salmon. Where we're going now is with a lot of automated sensors. So usually when we're paying attention to our farm and what's happening with our crop, we send a boat out. And that's expensive, and we take water samples, we look at nitrogen, phosphate, dissolvable organic carbon, we look at temperature, and temperature is very important, as well as salinity. I say this is part of a project called Impact, which is in its final year, it's EU funded. We have another project that's just started last week called Astral, and this will build on the sensors that we're using here. And this is looking at climate change, or the impacts of climate change in aquaculture, in particular seaweed. So you can see all the sorts of things that we monitor. Our site's being converted to what we would describe as an integrated multi-trophic aquaculture site. So we're introducing oysters. This is actually to help the economics as well, because at the moment, because we're not at a large enough scale and we're potentially not getting enough value out, the economics aren't that great. But introducing another crop to the same site, and you start to shift economics. Next slide, please. Oh, that's it. So where seaweed cultivation is concerned, and this is something that's not necessarily being addressed, there's a whole thing about how do I have a successful farm? How can I tailor my biomass to do what I need it to do? And it's everything from the site you select. So not all sites will be ideal for the seaweed that you choose. So there's a species and a site selection to bear in mind. At the moment, most people who cultivate grow a species called Saccharina latisma, or sugar kelp. That was pushed by biofuel and bioenergy. But it could be that that's not the right seaweed for you. There's also what we describe as the carrying capacity of the site. So how much material or biomass is a site going to be able to support? And this is in terms of the nutrients that are there before you start depleting everything and you end up with what we would describe as a dead zone. There's conflict resolution. So there's everything from navigation. So the marine environment's a very busy environment. So how do I stop a boat going through the middle of my farm and taking out my lines? And there's the communication and there's social license. There's also disease pests. It's a crop. It's agriculture. So it can have issues then. There's infrastructure, I say there's licensing, but there's also then the biomass composition. So when can I harvest? And if we're looking at things like carbohydrates, there's a major tipping point where carbohydrates are concerned. So the seaweed will start putting on carbohydrate content from about March onwards to sort of May, early June, and then it will drop dramatically after its main peak. So it's the window you have to be able to harvest. Can we go to the next slide, please? So this is what I mean about composition. And I think actually it's not showing on the slide. Emma, you just click? Can we get the rest of the slide up, please? That's it. So this is what I mean about, oh, no, back. This is what I mean about composition. And if you have a look, this is for Saccharina latissima. We actually have this data for 3 different kelp species, different brown species. And you can see what I mean about how you're getting a build-up and then a drop-off. Mannitol's very good for being able to follow what's happening with our carbohydrates. And this is the tricky one. It's developing those techniques that you can have a very quick analysis done. So as scientists, we like to science the hell out of things. And for me to do complete carbohydrate analysis, I take a sample, I freeze dry it, I then put it through an HPLC. So it's coming up with quick methods for actually doing the seasonal composition. We go to next slide, please, Emma. Where bioenergy is concerned, I'm very happy about this. This is a project that actually finished at the beginning of this year, and this is the macrofuels project. And basically 4 tons of seaweed was taken from SAMS. We managed to get it across Europe quite nicely because we dried it, and it was converted then into butanol and also ethanol, which were then added to a cart. Not that much, but just to make sure that you can actually drive on this fuel source. We actually were at the beginning and the end of this project. So we were at the beginning doing the cultivation and the environmental impact analysis, but then also at the end, taking everybody's byproducts and doing anaerobic digestion on it, which worked really well. Even with one of the thermochemical conversions that were involved, we thought that wouldn't anaerobically digest, but it was one of the best ones actually for that. So it can be done where energy is concerned, but what was obvious from this project is there's other stuff in the biomass when you do the pre-treatment in terms of using acids that interfere with fermentation. So you end up having to do dilution on the fermentation, the sugar that you're using for fermentation, and you get carryover things like pigments. So if you can get the more important stuff or the more high value out right at the beginning, and then do the energy after that, you're going to again start shifting the economics. Can we go to the next slide, please, Emma? So just to finish, I say most of our funding, I have to confess, has come through the EU on this. Things are starting to change in the UK. And it's been quite fascinating over the last 6 months, even in lockdown, the number of inquiries we've had from people, about 30 different small companies, businesses who are interested in setting up seaweed farms. What has become clear is that people aren't too clear about what to do with biomass. So there is a gap between people who want to produce the biomass and then where the biomass is going to go. So the link is there for the YouTube channel. So if you're interested in learning a bit more about what we do with seaweed at SAMS, please have a look. Thank you.
Thank you very much, Michelle. So I think the plan is that we will have questions and a discussion at the end of the talks. So Emma, if you can pass us.
I think Dan is going to switch in now and Patricia is going to present her own slides.
Yeah.
Okay. Yep.
So I'm going to share my desktop, which I'm hoping you can see it now.
Excellent, thank you, Patricia.
Good.
So,
And
yeah. Okay,
there you
so
go.
brilliant. No, that's up and showing fine. So the next talk is from Patricia Harvey, who's at the University of Greenwich. Okay, so thank you, Patricia.
Thank you. Well, I felt that it was probably right to talk about the microalgal side of feedstocks. Michelle and I are both co-directors of Algae UK, with Michelle wearing the hat for macroalgae and me wearing the hat for microalgae. So I'm delighted to be able to talk a little bit about the microalgal feedstocks. I think it's worth just putting in context in the the productivity of in Europe of algae in general, and it's tiny compared even to the USA. So, for example, about 1% of the worldwide biomass supply between 2006 and 2015 came from Europe. That was it. The rest, and the majority of that is is based on macroalgae. And it mainly comes in Europe from Norway, France, Iceland, and the Russian Federation. But the biggest market for algae, both micro and macro, is Asia Pacific. And that's come about due to the rise in population and the increased demand for food. So if I then move to look at positioning microalgae, these numbers were produced in 2014, and they have gone up just because of a massive drive to try to get more microalgae. But in 2014, the tonnage globally was less than 20,000 for all microalgae. And there were three main types: chlorella, spirulina, and dunaliella. And you can see the tonnages are tiny compared to macroalgae, which is at least 100 times more tonnage than microalgae. As I say, that's massively accelerating in recent times, in particular related to spirulina, which is now positioned as a major health food. So we're talking about 18,000 tons now. It has gone up a lot. But I just want to place, first of all, my own thoughts in terms of macroalgal biofuels and microalgal biofuels. And it comes from our own experience looking at the alga dunaliella, which is an alga that produces glycerol. And we set up, we got 10 million euros from a European project to look at a biorefinery based on dunaliella. One of the aspects was to explore the value of getting the glycerol out, because we'd worked with a company to convert an engine to provide combined heat and power for the science and engineering faculty at the Medway campus. And you can see an aerial view of that campus. But we also worked out that for 8,000 hour operation of that engine, we'd need about 1,800 tons per annum of fuel. And we then looked at the productivity of fuel from Dunaliella salina. 80% of the ash-free dry weight of that alga is glycerol, but we worked out we need at least a 50-hectare site operating 350 days per annum to get near to providing what we required. So in terms of scale, I think no matter what crop you're looking at, you have to be very, very mindful of the economics of how you're going to get there. And there have been a number of different reviews looking at the techno-economic value of microalgae. So production of microalgae for biofuels is not economically viable at the present time. Nevertheless, there are a number of different aspects of microalgae that are becoming majorly important and could provide the carriers, if you like, or when microalgae become utilized to combine or contribute to the biofuel agenda. And the industry trailblazers that are coming on board do come in from the food side, and they are demanding feedstocks. We've got a Unilever Algenuity partnership that was announced in July this year to explore the use of microalgae protein. And then last year in November, Nestle Corbion joined forces. So these are big food trailblazers, and they will be looking for large quantities of microalgal feedstock for particular components. And you can look at this slide, which shows what we're talking about, the protein, carbohydrate, and lipid percentages compared to soya. And you can see that of the major microalgal types that are being produced at scale, they've got a pretty good amino acid profile. They've got plenty of carbohydrate and starch, and the fatty acids, including DHA and EPA, polyunsaturated fatty acids are prevalent So in terms of looking for sources of food ingredients, it is not surprising that the companies have started to look at microalgae, but they will need scale and reproducibility. And here is a slide showing just briefly some of the major microalgae that have been produced and what they're being used for, either as a whole alga feed up until recently for fish feed, or for extracting high-value molecules like beta-carotene, astaxanthin, phycocyanin, EPA, and DHA, leaving biomass that needs to find a home. And indeed, one of the major areas of these microalgae that has been taken up in a major way have been based on natural colorants. And you can see here the Dunaliella yellow-orange color, Spirulina blue color, and the Haematococcus pink-orange color, which come in as food colorants because they're natural, and there's a huge market for those that's growing. So I want now quickly to touch on to what I think are the 5 main reasons why industrial production of microalgae is not yet well developed and really needs to take off. We've got limited capacity and know-how to cultivate microalgae at large scale. There's also sustainable low-cost methods needed for harvesting. And I'm going to pick up on each of these briefly shortly. And then there's pre-treatment and processing methods are needed. There's a lack of knowledge of the food ingredient value that you're going to get out of these microalgae, and then there are regulatory considerations. Off on the cultivation side, we were lucky enough to work with one of the big majors producing Dunaliella based in Israel in the project that we had. And what became clear is cultivation know-how at large scale. Really, it's all about farming. We've got different vessels that you can contain microalgae in. Open pond raceways shown at the top. To the side of that, a cascade type raceway to contain the algae. These are both open systems. Then you've got contained photobioreactors where a lot of effort has been placed to cultivate microalgae for oil for biofuel in the past. And then a picture depicting some of the key elements that matter in terms of farming in general. Carbon dioxide. Where are you going to get your carbon dioxide from? In these systems, we're using flue gas CO2, which is ideal for algae. Nutrients. You've got to get your nutrient balance right. Choose your inocular in order to be able to improve your biomass level. Get the light right, the temperature right, and also manage rainfall. So it's a farming game. And in terms of yield, we expect from microalgae to get about 25 grams per meter squared per day. Our actual yields in this project were from 0.75 to 4 grams per meter squared per day. This is ash-free dry weight, not getting salt on it. But we also worked out that if we could increase that productivity, which we think we could, to by 83%, we get a 27% profit margin on production and an internal rate of return of 296% just by improving the productivity. Moving on to the next one, some of the other issues that dog all crops, farming, and it applies just as much to microalgae as any. There's an aspect of managing the pests that suddenly appear and also managing seasonality. And you can see the top right-hand image, bags of microalgal powder that have been juiced, but they're different colors coming from different seasons. The next one is this sustainable low-cost harvesting. For microalgae, the cells are small and in low concentration, and you need different solutions for different types of species. With our particular alga, we have a wall-less alga in the form of Dunaliella. You can see the bottom picture there. It's got no cell wall, it's got an outer membrane. We worked with a company called Evodos to harvest the algae, and we were able to get 23% solid paste harvested with their technology, with 90% of the cells intact, but it came at a cost because the rate of harvesting was 3,000 litres per hour to get that, whereas the industrial processes using Distat centrifuges was 12,000 litres per hour, but they tended to rupture the cells. So which way do you go? And there's a huge amount to be done in terms of harvesting these algae. In terms of processing algae, or getting out the high value ingredients that you might want, leaving your residual biomass, biorefining costs about 20 to 40% of the total production costs, but they may increase to up to 50 to 60% of the cost. And it's also important to get the scales right. So if you're going to extract carotenoids and leave residual biomass, you better make sure you've got the right amount of material being produced to match the industrial facility that you're targeting. And also understanding that whole algal biomass behavior. We nearly set one of the systems alight because the filters got blocked. And then there's also a knowledge of the ingredient value that you want. And so in terms of being able to really understand which way to go with microalgae, if you're targeting an ingredient or a food additive, then you need to have an understanding of what the European market is about. And until you've got some biomass to produce, it's very difficult to start to work that through. You can see here just increasing trace amounts of algal processed residue being added to sausage and to bread by one of our partners, which affects the texture and also potentially the taste of the food products. And of course, we don't yet know fully what the European market would like. And so, in fact, there are various other spin-off side projects, and I put this slide up because we have a project, an Interreg project looking at valorizing algae for taste. But it also reminds me that we have an online event on the 28th of October, 10 o'clock to 12 o'clock, developing the UK roadmap for industrial applications of algae for food and novel food ingredients. If you want to attend that, please get in contact with k.covington@ucl.ac.uk. And then the last consideration are the regulatory considerations. So we've got food, feed, nutraceutical, cosmetic ingredients, all subject to regulatory considerations. In Europe, we're subject to what's known as the Novel Food Act. It's European Food Safety Authority. Food that has not been consumed to a significant degree in the EU before May the 15th, 1997. All the food applies. Now, in terms of microalgae, food or extracts that were on the market before 1997 were only 3. And since then, the Novel Foods Register, since 1997, there have been 5. And part of that is this huge, massive weight of documented evidence that's required in order to be able to get these products into the food chain. So it's not an easy game to be able to handle these novel algal feedstocks to get them into the marketplace. It also applies to feed. Feed regulations are probably more onerous than the food regulations. They also apply to fish, to zoo, to circus animals, creatures living really in the wild, all feed material and all colouring agents and nutritional components. And then in terms of extracting novel carotenes, for example, if you want to have a nutraceutical claim on a particular ingredient from an alga, you need a medicine license. And that carries, again, another big burden in terms of cost to be able to get it into the marketplace. And we're wanting all of these to try to drag the feedstock production up. And one of the last areas is phytoene and phytofluene, which are colourless carotenoids for cosmetics. And to get those into the marketplace, you need to meet REACH regulations in Europe. And I think quite a lot of these regulations are still going to apply within the UK. Even after Brexit, although I'd like to suggest to everybody they just bring everything to the UK. But you can see that there are a number of different regulations that have to be met. So to summarize then, I believe that in the microalgal field, there is a real need for improving our productivity and both, for scale up and in scale up. And then also developing the handling know-how, both for being able to process the material and to produce products in order to be able to then manage the totality. It's also really important to get sustainable production going. Many systems are being set up to cultivate microalgae using flue gas CO2 in order to be able to reduce CO2 emissions, and that's a very good way of being able to go forward. But also many of these algae grow on non-potable water. Dunaliella grows on salt water, for example. And of course they use solar energy. And I think another really important way to go forward is to really consider a biorefinery approach where nothing leaves without an invoice. And then to complement that, it's incredibly important to get that market development sorted out. We've got to work out what makes a tasty product and what gives health benefits, and also get a driving force for legislative approvals. I just want 2 more final slides, and that is that Dunaliella salina is being cultivated in these little mini pond raceways and very big raceways in the middle of Spain. And this photograph was taken in the snow. And the algae was growing extremely well in the snow. What we have here is an inland salt mine where the salt water is being diverted to cultivate, or some of it is being diverted to cultivate microalgae. The whole operation is a salt mine, and CO2, natural gas, is combusted in a CHP engine, and a 20 megawatt CHP engine to provide heat for salt production. But the CO2 flue gas is an emission which is not wanted and is being fed back into the microalgal ponds. But on top of that, heat from the CHP engine is being diverted to make sure that the ponds are kept warm, because the value of the microalgal has started to go up massively. So cultivation in snow is possible. I would like to think that we also might be able to cultivate these algae in the UK, and there really is a big need to get scale in the UK. Thank you.
Right,
I shall
thank
try to unshare my screen.
Brilliant. If Dan can, fantastic. So Dan's got the next slide up. So as I say, if we can save up the questions and the discussion for the end of the presentation. So the next speaker is Jessica Adams, who's at Aberystwyth University.
Thank you, Ian. Doesn't say I'm on live. Yeah, there we are. Hello. Thank you for that. Yes. So I was going to sort of talk more on my aspects, which are a little bit more in the middle between that which Michelle and Pat have been talking about. So the next slide, please. So I first got involved with Supergen back in 2007 in a Marine Plus project, which is quite different to the current Supergen Marine. And it was, but this project was looking at making biofuels from seaweeds. So what I was doing is I was going out, I was collecting some material once a month over a year, and then using it to produce bioethanol, biomethane, and then I went to Leeds and we did some pyrolysis, and we made some pyrolysis oil from that as well. And that sort of got me really interested in the seaweed, and I've had the interest ever since. It sort of had to wax and wane a bit depending on sort of funding, etc. But it's now I've got sort of 4 areas I've got focused here. So the first one is scaled up processing. So in Aberystwyth, in that previous slide, you can see our old processing facility for a project called Beacon, which is now moving into our innovation campus, which got finished at the end of August. So we've got these pilot plant facilities, which are just amazing because it just gives you the intermediate step between bench scale and industrial scale. And also with that, I've got involved with a number of different industrial collaborations with companies working on seaweeds at a number of different levels. So I suppose a lot of this is echoing what we've just heard, but I think biorefining, producing multiple products, has got to be the way forward, particularly for seaweeds. You know, you need to be looking at not just the one product, but also what you class as waste or residues. What else could you do with it, including things sort of for lower value things through fermentations and anaerobic digestions, as well as taking out your carotenoids or phenolic compounds. I've been doing some work looking at plant growth trials over the last year. So this is sort of looking at seaweed for fertilizers. And then I've got an interest looking at novel enzyme discovery and characterizing that. I don't know how relevant that is to the Supergen program, but that's also there. Next slide, please. So I've got a number of different relevant projects that are going at the moment. So in the top left-hand corner, there is this a mill that we have brought in. So we've got some contract research going where we're processing 900 kilos of seaweed. We started this morning. It's all going very well so far, which is very pleasing to know. But yeah, so that's producing a number of different compounds from the seaweed. In the middle picture is our pilot scale steam explosion unit. So you can put in about 5 kilos of material into this, and what it will do is it will heat it up with steam and pressure, and then will reduce the pressure very rapidly, and that will rip biomass apart. So you typically use this for sort of more lignocellulosic, for woody materials. But last year we did a trial and we put in some seaweed, and we've also done some water hyacinth, the marine algae, the marine plant, to see what the residues look like. So this was done with Leeds University, particularly with the Bethwan project led by Andy Ross there. And we were comparing steam exploded material under certain conditions with hydrothermal carbonisation done in the same kind of conditions, and then looking at the product and what you could get out of it afterwards, what level of inhibitors, etc. there were. The third picture on the top on the right is just some other work that I've got going on. So this is some data from one of my PhD students. So she's got a drone and she's looking at algal blooms that are happening down in the Milford Haven estuary down in South Wales. So these are blooms of green algae that happen every year, and you can see basically that using the red-green scale here for a particular type of camera, which basically measures your vegetative index, sort of how green it is, basically, that you get a lot of fluctuation within an area across time, and also within the estuary itself. Down at the bottom left is some of these plant trials. So we've got another plant trial that's going to be going in next month. So we've got some fertilizer trials. So what you've got is basically you can take photographs every week and then use computers to analyze it to work out the surface area and the diameter of these plants. And then at the same time, we're going to be running a char program. So the bottom right picture is a bit of a cheat. So this is our half a ton per load char pyrolysis rig. But I'm not actually going to be using that. I'm going to be using a smaller one, again, to produce char from seaweeds, which we'll be using at different levels to see how they're going with the plants. Next slide, please. So the number of kind of products that we produce from seaweeds, it varies considerably. So we've got things like fuel. So we've made ethanol and we've made methane, but we've also made some interesting molecules. So the top picture is showing the phenolics. So all the phenolics are produced from this molecule shown there called fluoroglucanol, and you can see that there is lots of spikes with regular intervals between them, which shows that you've actually got a polymer of these phenols, with that gap in between them being the size of that fluoroglucanol molecule. And underneath is some fucoidins. Again, so we've done a bit of work on the fucoidins, so those are both from the brown seaweeds. And then on the right hand side, we've got some solids. So in a previous project, we actually produced some pellets from seaweeds, and then they went through some specialist pyrolysis system and we produced some char, which you see at the bottom. At Aberystwyth, our job was then to involve it in some pot trials to see how well plants grew with a small amount of this char in it. Basically, they didn't, is the short answer. But I would very much stress that all chars are not equal, and that I think there's a lot of work in this area that still needs to be done, because the primary issue which I've come across over and over again is that people treat seaweeds as if they were terrestrial biomass, and the composition and the way that they act in situations is very different. Next slide, please. So just some more products. We're also producing a number of different liquid extracts. So the image on the left is showing a membrane filtration system that we've got. So basically, if there is a particular fraction from a seaweed or from anything else that you're interested in, you can use membranes and you can take off, for example, material that is much larger than it, and you could also take off molecules which are much smaller than it, and then you can concentrate up the fraction that you're interested in, which I think is a very useful method for separating products from seaweed and beyond. In the middle, we've got some film. So this is another bit of work by a PhD student, and they're looking at producing films from algal extracts. You can see the one at the top is brown very clearly, but if you look carefully, there's also one underneath. So it's looking at different properties as well as what the films actually look like. And then lastly, I've just begun doing some animal feed trials using seaweed. So we're looking at methane effects there. And sounding very greedy, I think there's an awful lot of work that can be done on lipids and proteins as well from seaweeds, from the extracts. So next slide, please. So these are what I have sort of identified as sort of research priorities. Obviously, this is part of the debate following on from this, but I think, as ever, I think flex funding calls should be as interdisciplinary as possible. And as I've just sort of mentioned before, I think we should be looking more at the novelty between your algae and your terrestrial plants. There's seaweeds have got greater variety there than all the terrestrial plants put together. The composition is different. You've got different carbohydrates in there. You've also got carbohydrates which aren't there, which you typically find in terrestrial biomass, and all this has a big knock-on effect on the storage. And things like, you know, your ash content, there's a lot of sort of expectation that ash is going to be similar to that for terrestrial biomass, and it really isn't, because the seaweeds take up so much of the elements in water. So there's a lot of looking at the processing and whether you might need to do something differently for seaweeds than you would for terrestrial biomass. And then sort of following on from that is also for marine plants. If you're working with material that's come from a watered environment, then you are going to have to use different systems. It might be that they have much higher water content. You might need to drain them before you use them. There's a lot of different things that you need to think about for processing algae, which you wouldn't necessarily be thinking about for terrestrial biomass. And this then follows on to the cultivation, which Michelle was touching on at the end. I think, again, there's a lot of work that's done on the cultivation, and there's a lot of interest downstream. I too have had loads of companies interested or wanting to work in this area. This is definitely a growing area, but I think there's still this gap in the middle where we need to be looking at downstream processing. And on a very personal level, where we were using the mill the other week for this contract research, we found that you don't need very many stones or very hard shelled snails to go in there before it starts denting your mill blades. So again, is there other things that could be done? Is there, if we say this is a major issue, is there any way we can make sure that we actually remove more of those snails before it goes in there? Again, this is the kind of thing that you need a two-way dialogue in order for things to happen. And then finally, I've got life cycle analysis and technical economic assessments, because I feel that it's all very well us coming up with these brilliant ideas, but if they are utterly unviable, I think it's better to know that sooner rather than later, so you can actually work out what is a more viable process and then start focusing that way. So that is the end of my slides. So thank you very much.
So thank you, Jessica. And then on to the next speaker, who is Ian Watson from the University of Glasgow.
Hello. The first question is, can you hear me okay? Because I've had problems with my microphone.
Yeah, I can hear you fine, Ian.
Excellent, thank you. Okay, so my next challenge is that I had some confusion over my time. I thought it was 10 minutes, but that was if there was time available. I have 2 minutes, I think, maybe 5 in the schedule. So I'll ask if you can move to slide 4 then, please, Pam. I missed some of the preliminary introduction. So hit again, please, thank you. So I think one of the major problems is really looking at what the bottlenecks are, trying to address solutions to that, and in the next couple of what those are. So there's a couple of seminal papers by Christie that looks at this in detail, and they're certainly worth reading to understand what the problems are in terms of processing and the inputs and the outputs for micro algal biorefiners, for example. So obviously on the input, we've got light, sugar, nutrients, CO2 and water, and all of these are potentially expensive. a lot of the research that we do in Glasgow is looking at trying to minimise the costs associated with these. Next slide, please. And hit again. Thank you. So on the downstream side, there's obviously a lot of cost associated with the separation of the microalgae and what you can do with the actual microalgae once it's produced. So particularly looking at extraction of high value products that have been mentioned already, and then what feedstock you have left from that in terms of utilising it for energy purposes. Okay, next slide, please. So if we look at the general outline of a biorefinery, hit again, please, maybe a few times, maybe 3 or 4 even. The joys of animation when you're not in control. So it's essential that you have a sustainable feedstock, so microalgae fit that criterion very well. And reducing the energy inputs to the system is critical, and also water. And we're looking at some various aspects of that at Glasgow. In terms of the outputs on the right-hand side, high value products, as mentioned, liquid fuels, and also what you can do with heat and power, whether you can use any of that feedback. It's interesting to see the work being done on the salt mine. Hit again, please. And obviously feeding back CO2 into the growth of the feedstock, which is really sort of a common area. So for the next slide here, we can see what I quite like this slide. It's a graph of the representation of the lipid profiles you can grow through outside raceways, for example, globally. And I won't dwell on that. References there for those who are interested. Next slide, please. And the one after as well. I'll quickly give you an overview of some of the work we're doing at Glasgow. We focus on all aspects from growth down to refining, product development, and also fuel. Most of the extraction we've done, we've looked at solvents and trying to enhance solvent extraction through microwaves, for example, and also some thermochemical processing, which I won't talk about today, but the extraction of the energy and valuable components is an essential part of the overall economics. And as has been mentioned, the life cycle analysis and PEA and analysis are all contributors to that process. We work on novel photobioreactors. We're trying to compare those with outside raceways. So we're collaborating with people in Colombia, for example, and also in Japan. And a lot of what we do is also looking at real-time control of photobioreactors, so pH control, when do you inject CO2, for example, and all these issues, and looking at relatively simplified or inexpensive, at least, let's say, a control system. Okay, next slide, please. So let's go through these fairly quickly. It's just a 6-tube photobioreactor we have there, and data login and data control for that system. Next slide, please. And just some of the system components in that. Next slide. So you can see we have LEDs on the base. Next slide, please. But also, we look at IBS tanks. It's a very inexpensive way of going up to the cube, and we've done modelling in terms of the light distribution within that process. Here we can see some of the sensors that we've looked at developing. So image processing of images captured through microscopy, for example, looking at flow cytometry as well in that, also image processing and fluorescent staining and Raman spectroscopy as a way to try and identify the point at which you want to harvest as well, but also at what point you want to inject nutrients and CO2 as mentioned into the photobioreactor. Okay, next slide. So here we got just various vacuum filtration systems. We've looked at organic flocculants as well, and also some novel pump designs that require 0 to very low amounts of electricity to operate in order to filter out the algae very efficiently. Okay, next slide. This is probably too detailed, but looking at different lysing methods as well. So how do you actually crack the algae? Some of them have very hard outer membranes. So here we're looking at Maltopsis occulata, for example, and comparing and contrasting different methods of cell lysis. And that was looking from lasers up to a water bath. While the laser was very efficient at lysing practically all the cells, it wasn't practical for large scale volumes. It may be useful for a lab-based system, but not necessarily something for industry. Okay, next slide. And maybe the one after. So we also looked at the effects of hydraulic pressure, for example, and also coupling that with liquid nitrogen. Not necessarily a cheap way unless you have processes there where you have cryogenic cooling already on site. But there you can see contrasted differences between just the liquid nitrogen and also with pressure. So probably about 30 bar. The impact of using liquid nitrogen was negated, and there was no difference between that and just applying pressure and also temperature as well. So next slide. Next slide, that's just looking at solvents. And it's important to characterize what lipids you're producing within the algae. Each of those potentially have a higher value. And in terms of utilizing them or going on to producing something else downstream, more energy related. So here we're looking at using microwave to try and enhance the extraction process. And interestingly, it's not just a case of increasing the power. We had a higher extraction with the lower microwave power than with the higher microwave power. So there is an optimization process involved with that. Next slide. Okay, let's skip a few like current work just very quickly. So this is just a quick outline of some of the PhD students I've currently got working. So Abdulaziz, he's looking at production of bioplastics with Chlorella vulgaris. Next slide, and the slide after. So skip 2 slides, please. Talupe, he's particularly interested in the role of different carbon sources on increasing growth, and we got some interesting results there. Unfortunately, I don't think I've got time to go into, but next slide. Tanya, I think this is a strong way forward where you're looking at microalgal biofilms. So the key advantages here is the dewatering step. So if you're growing the algae in very limited water, obviously the dewatering costs have really reduced. We've got some good growth on different films here, different substrate materials. One of the problems is looking at trying to monitor growth consistently and accurately over the surface area that you're currently growing the microalgae over. Okay, next slide. And Nadine, she's looking at the benefits of spirulina on brain function essentially, and also the behavior of children, for example. So they were collaborating, as I mentioned earlier, with the team out in Colombia with a view to also contrast growth performance with photobioreactors and race rates. Next slide. So that's a very quick whirlwind tour of what we're looking at, and I'll pass you back to Ian.
Thanks very much, Ian, and also thanks for going through that so quickly. That's great. Excellent. So the final speaker we've got this afternoon is Maulud Ouadi, who's at the University of Birmingham.
Yeah, so we heard a lot about the challenges, some of the main challenges about algae. So what my presentation is all about is how can we try and start addressing those problems and start piecing everything together. So next slide, please. So this was a piece of work we did with Harper Adams, Aberystwyth and University of Durham. So there's a big problem with seaweed. It gets caught up in fishing ropes from fishing vessels. So we tried to address this by converting it into a biochar and fuels for marine using thermocatalytic reforming at a small scale. Next slide, please. So one of the things with seaweed is it's extremely difficult feedstock to collect and handle. It has moisture contents of over 90%, which means you need a very high drying efficiency, and you need a lot of energy for the drying in order to process it by thermal conversion. It's also prone to rapid degradation. So once you collect it, if you don't ensile it very quickly or have some way of preventing oxygen to get to it, it goes off very quickly. But once you do dry it and pelletize it, it forms very stable pellets which can be used in downstream gasification and pyrolysis technologies. Next slide, please. So what we did was converted the seaweed, and we got 3 major products. We got an oil around 6%, gas around 36%, and a char around 37%. The bio-crude oil we obtained, we compared it directly to the marine ISO standard 8217, which is the standard if you want to use it as a shipping fuel. So you can see the parameters with our oil, which is TCR compared to the standard. We're not too far off the standard, but no matter which technology you use, which pyrolysis or hydrothermal liquefaction or whatever you're going to use, you're going to need some degree of pre-treatment, but the extent of the upgrading treatment, usually by hydro treatment, is not that much. Okay, so there's some issues to address with optimization of oil yields. How do we avoid drying? How do we optimize oil? But alternatively, do you really need to go for oil? If you want to go for marine fuels, you can also use gases. So there's a lot of ship engines now running on gas. So it's a selection of which fuel you want to target. Next slide, please. So this was the gas composition. You can see our gas is around 19 megajoules per meter cube. So that's a good calorific value for a gas. It has a very high hydrogen yield, CO yield. You can either use that gas directly in the ship engine as it is, or you can use the hydrogen. You can extract the hydrogen and use hydrogen for ships, or you can turn the hydrogen into methane and LNG for ships, which already exist. Alternatively, you can use that hydrogen to extract and upgrade the bio oil itself, because the upgrading process requires hydrogen. The residual char is full of ash, it's 73% ash. So in terms of energy, it's not such a good vector to use if you want to extract any energy out of it, but it still has a residual calorific value, which you could perhaps look at co-processing with other biomass in a gasifier, for example, to produce the heat and power you need for any process. Next slide, please. So this is one of the concepts we was thinking about. So this is, I mentioned to you before that this seaweed is really available all over the place. It's not unless you're farming seaweed or you're farming algae, this is going to be all over the place. So what you really want is to be processing this close to the source of collection. So a concept we had was basically that these fishing trawlers, when they collect the seaweed off their nets, it goes directly into an onboard reactor. This is then converted into a biocrude. That biocrude can then be taken to a central facility at the port, which can then be upgraded. So the fisherman would get a small subsidy for his oil. That would then all go into a central refinery, which would be upgraded into a marine fuel. Alternatively, you could use heat and power on board these ships. So a lot of these ships are power plants in their own right. They've got a lot of heat and power available, which you could use from the boilers, for example, which you could use to dry the biomass. So once you do that, you can then put it through a process, for example, pyrolysis or TCR process. You can extract your gases. Those gases could be used directly in the ship engines to fuel the boat. So next slide, please. So one of the things we need to think about is different systems. We heard a lot today from the other speakers that this needs to be economically attractive. So the only way you can really do that is if you limit the amount of processing steps. So there needs to be a techno-economic analysis around all these different systems to see which one's the best in terms of economics. So I've highlighted a few here. So the top one's the process we've already explored. So it's collecting the seaweed, ensiling it, drying it, and then pelletizing it through a TCR. There you get your syngas, you get your bio oil, which can be upgraded, and you get your char, which can either be used for fertilizer, but most likely it would be used as a gasification medium. So the challenge is in that red box. You want to basically remove everything in that red box. And a feasible way to do that would, if you're, for example, if you're farming algae, you could do a lipid extraction. We heard a lot that there's a lot of value in some of the chemicals produced from algae. So you would extract those first. The residue would then go into a hydrothermal liquefaction reactor. That doesn't need drying, you can use it wet, it just needs pressure, maybe about 300 degrees temperature. Your yields of oil would be about 50%. That would then go into a hydro treatment process to get your, if you want liquid marine fuels, that's what you'll get at the end. But then you have to consider, where does the hydrogen come from in that process? So you may have to combust some of your char in a gasifier to produce that hydrogen, or use electrolysis. The final route, which is a hub and spoke model, would be the most likely route moving forward to avoid transport costs and storage. You would get your seaweed from the fishermen would get their seaweed, they would convert it into biocrude by hydrothermal liquefaction on board the ship. That oil would then be transported to a central facility at the port, and then upgraded using hydrogen there, and then converted into the liquid marine fuels. So there's just some thoughts around the different process models, but this needs to all be investigated and looked into in terms of techno-economics. Next slide, please. Yeah, so this is really more of the same, just that where is the seaweed in respect to these key port terminals, how much is available, and then we need a techno-economics around the whole concept. And then by doing that, you'll be able to strategically place where you want to have your central depots, where you want to, which vessels you want to target. And I mean, we had inquiries from naval people with naval ships who were interested in putting onboard facilities on naval ships. So it's a question of which ships do you go for, which ships have power, have enough heat to do the processing, and then you can start developing your model like that. I think that's the last slide, Emma. Yeah, so thank you.
So, thank you. Right. So thanks everyone for the presentations. There's a lot of information, I guess, in a short period of time, and I guess does indicate the quite a range of activities that are already taking place in the UK. I guess what we're particularly interested here is thinking about what a call might look like related to this in terms of algae, and how that might relate to the activities that were described at the beginning that is already taking place in Supergen. So Dan, were you picking up the questions at all that may have been coming in or are starting to come in?
Yes, so we've been reviewing the Q&A and we're waiting for questions to come in. Some have been answered, but it doesn't look like we've got too much activity at the moment. So if anybody does have a question, please do submit them.
Ian, there was one question earlier on. Will any research proposal on algae feedstock for food application be eligible under these funding? I'm not exactly sure about that, but I think if it's a food application, it's probably not eligible within the Supergen remit, but you may have an opinion about that.
Yeah, no, I mean, and I think that one of the things we were hearing about today is the way that it may be, you know, in terms of for the overall economics, it may be important to actually have whether that's, as we were hearing earlier on about, for example, co-cultivation with actually with oysters, you know, in terms of with the algae, but also in terms of, I guess, the biorefining of the algae. But I guess clearly this is about power in terms of, or energy production in terms of within Supergen. It is an energy theme. So really, you know, there could be food within it, but I think there has to be a significant element of energy, and it's just that the wider system includes food. Would be my take. I don't know whether Patricia wants to say anything else, but I think it's very difficult, even with land-based energy, to completely devolve it from food production, in terms of, we obviously use land for food production, but I think clearly the focus of this has to be on energy.
I think Patricia Thornley has already had to leave the call, actually, and so she won't be able to comment.
That's fine. It's just if there was a, but
yeah.
that would be my take. I think it needs to. So yes, I think a wider system, and that may help with the overall economics, and that's clearly important. But the focus does need to have a strong element of energy in it. So were there any other questions?
Question, Ian. So this call, it's a call for £100,000, and two are going to be funded from the same topic, or is that across all three topics?
So I guess MMA, which
yeah.
Yeah.
So yeah, it's across all of the three topics, and in total 2 projects will be funded. We're not going to say in advance which, we're not going to judge in advance. It will be purely on the merits of what proposals are submitted. So if we get 2 really good calls in 2 really good proposals in one area, we may well fund 2 on one area and none on the other 2 areas, but it's going to be an open competition, really. Okay. And the projects will be 100,000 at an 80% FEC basis, so 125,000 in total, but you'll get £100,000 as your funding.
Is that just from one institution, or does it have to be collaborative?
We do prefer the projects to be collaborative projects if possible, please. Yeah.
So in the Q&A, we have a couple of questions come in. One for Emma. Can you describe collaborative institutions, for example, academia, industry, the CPI?
Okay, well, we're happy for anybody to be a partner in a project. We generally can't fund industrial partners, but we are able to fund academic partners, and your co-partners do not need to be members of the hub necessarily, but the PI needs to be a member.
We also have a question for Jess. Says that someone is interested in your other molecules of potential value. Can you describe them a bit? Professor Robert Nash in Aberystwyth researches natural products from terrestrial plants, specialising in amino sugars.
So I've mostly been doing some work on laminarin, which is the main polymer in your excuse me, on the brown seaweeds. But we have got some interesting extracts which we've taken, which still need to go through the LCMS, the chromatography gas mass spectrometry, to actually sort of see. But without wishing to sound like I'm dodging this one, possibly I could chat with him outside the session.
Okay. Have you seen the question on the eligibility one, about somebody who's got 5 years of postdoctoral experience and is asking about eligibility to apply?
Yes, I have seen that. I think I know the postdoc in question, actually. I think he's eligible because he currently works for oh, if it's a fellowship, then I'm not entirely sure, actually. I'll have to get back to you about that, Santos, to be honest. I'll check the rules on fellowships, because I think there is a limitation on how many years experience you have, but we may be changing that anyway. I will check that and clarify it for you. But in terms of eligibility as a hub member, Imperial are members of the hub, and so you would be able to apply in that respect.
Question, Emma.
Yeah.
This collaboration, are international academics eligible as well, or can they be partners?
Yeah, we don't have any problem with it being international as well. In terms of the funding goes to the PI on the project, and then you distribute it, but you distribute it to your partners.
Okay, so that academic institute can be abroad, yeah?
We wouldn't be able to pay the money to an overseas institute, but if you get the project, I think you probably can. Again, I might need to clarify on that one, Malid, sorry.
Okay.
I think we probably need to check on the eligibility, because normally UKRI funding is for research in the UK, but obviously there will be specific rules in relation to, because of this is flex funding.
Mm.
What about subcontractors? So if they couldn't be partners, could they be a subcontractor?
Yes, I think so, yeah.
Okay.
We also have a question for Milou, re onboard facilities. Was this a drop-in fuel, or will it require system modifications?
On board, yeah, sorry, go on, Dan.
Also, have you considered the RSS Sir David Attenborough?
Yeah, I mean, no, I haven't considered that, but the fuel on board a ship, there's different models we can take here. If the ship has enough waste heat from the boilers, we can use that to do the drying of the seaweed, because that's the biggest energy demand. And then we can produce both a liquid and a gas, which can go into a dual fuel engine. You wouldn't need modifications of the engine, so we could use that directly. But if you want to avoid drying, then I would propose, for example, you have a small shipping vessel. I would propose then you do a hydrothermal liquefaction that's taken to port for upgrading our central facility.
Right, thank you. So we've got another question around eligibility, and specifically, I guess, the involvement of a industrial partner. I think from that question, the indication is, in effect, can there be an industrial partner, and it's just the academic partner who receives the funding. Is that your interpretation? Yeah, so here we go. There's Emma's. Yeah, so you can't directly fund industry, but clearly there would be added value to the project if you got industry involved, and particularly if they were putting resources in,
yes,
in some way in there.
We quite often have in-kind involvement of industrial partners, for example, who will give their time or their maybe even sponsor the project partly as well.
So if you're able to have that, then clearly put that and build that into the proposal.
Yeah,
and highlight that.
So, there was a question from Patricia Harvey, I think. What percentage of, for the seaweed producers, what percentage of fresh weight is organic and what percentage is ash? Or is that a response? I can't tell whether that's a response or a question.
That's a question.
Sorry.
I'm curious because certainly looking in terms of organic matter, which is what you're after. We at one point also looked at taking microalgae and using it on board ships to provide combined heat and power. But the amount of material, organic matter, that would be required was quite large. And I know that seaweed potentially could provide some really nice opportunities. But I'm curious to know what percentage of salt or ash people are generally finding in seaweed. I think it can vary quite massively and can be very high, and to what extent that would be a problem for engines.
For our seaweed, we got 35% ash on a dry basis, so the rest was organic. Well, once you've dried it, you've converted it. The ash obviously remains in the char, and then what you have left is just your products for the engines.
So just to follow up on that, it varies on the species. So you can get down to about 25% ash. So it just depends on the species, but there's also a seasonality to it as well. So sometimes the year you have a higher ash content than you want, but there is the possibility with some of that content that there's some useful stuff in it in terms of other metals that are present and how those are treated.
I think you're absolutely right, and one of the big things that we've been finding with sargassum is ash contents of 55% or more. I mean, it looked like it must have been some of the areas that this particular sargassum had been collected must have been grown on a volcano that had erupted in the ocean, just because there was so much ash in the seaweed. So it's very, very, very dependent. Maybe that's something that's needed, to get looked at carefully, dependent on the environment in which it's absorbing or mopping up all of its metals and its ions.
And it is age related. So, for a lot of the studies that have been done, you're looking at what we would describe as wild material. So it's not controlled, and you've got a variety of ages. As it ages, so something like Laminaria digitata, which can get to 7 years old, it usually has a higher ash content because it's as it's growing, it's carrying on taking those minerals in. Whereas something that's maybe 6-7 months old has a lower ash content.
Right, so it looks like we've finished with the Q&A. Doesn't seem to be any more questions coming in, and we have run to our end time. So I guess I'd just like to say thank you to all the presenters for presenting today, and thanks to everybody that submitted a question. We hope you found this webinar useful, and please do use the contact details available on your screen to get in touch with people if you need any more information regarding the call. Thank you, everybody.